Hello, welcome to ChemBio and to A-Level Biology revision. In this video we'll start looking at nucleic acids, and this is the last section in the biological molecules chapter. Stay tuned. There's one nucleic acid that certainly everybody knows about or at least has heard of, and that is of course DNA. In fact, the last two letters, Na, stand for nucleic acid. Oh, what a surprise. And DNA is a very important molecule because it is the basis of heredity in many organisms. That's to say that DNA is used to transfer genetic information down the generations. Now, nucleic acid, why do we call it in this way? Well, first of all, nucleic comes from the word nucleus because it was originally discovered in the nucleus of a cell. Nowadays, we know already that, of course, DNA is found not just in the nucleus but in other parts of the cell. And it's an acid because of the structure, and you'll find out why exactly in just a few moments. So we've seen already with previous biological molecules, such as carbohydrates, for example, that they are very often polymers. For example, starch. Now with polysaccharides, the structural unit, the single structural unit, the monomer, is of course a monosaccharide. With proteins, it's an amino acid. While in nucleic acids, the monomer is a nucleotide. We can say therefore that nucleic acids are polymerized nucleotides. Yes, so we have many nucleotides all joined together to form a very long chain, and this long chain is what we refer to as a nucleic acid. Here's the general structure of a nucleotide. Now this is just a diagram, I've only drawn this schematically. In the middle we have a pentose sugar. Again, back from carbohydrates, you should hopefully remember that a pentose sugar is one that has 5 carbon atoms. It doesn't matter what shape it is, whether it's a pentagon or a hexagon, whatever, the key point, there are 5 carbon atoms. Pent, yes? On the left we have a covalent bond with a phosphate group, and on the right we have a nitrogenous base. And to form a nucleic acid, quite simply, these nucleotides, there are several of them. In DNA, for example, fun fact, there are anywhere between tens of thousands of these nucleotides up to millions of them. So it's an extremely large molecule. It is the largest molecule that an organism can synthesize independently. But anyway, yes, these nucleotides are arranged one after the other to form a long chain, and they're joined together by another covalent bond. The specific name that we have to know for this bond is called a phosphodiester bond. Okay, phosphodiester bond. What does all of this mean though? Let's take a closer look at a nucleotide, and here is the structural representation of a nucleotide. We can see that on the right hand side we have a nitrogenous base. That's what the pink coloured shape was on the previous slide. It's quite simply called nitrogenous because it contains nitrogen. Other than that, as you can see, there are these rings, and these rings are carbon rings with nitrogen atoms inside them. And very often we can just omit the word nitrogenous and we can just say base. That's perfectly fine. On the left hand side, as we saw already, we have a phosphate group, and in the middle, of course, we have a pentose sugar. Don't get bogged down by the term phosphate group. All it means is that this is a functional group. If you remember from GCSE chemistry, that functional groups can be found in organic molecules that basically determine what type of molecule we're looking at. For example, with alcohols, the functional group is OH. So if you see that a molecule has OH at the end, that is what it distinguishes it from other molecules. That's the giveaway. It tells us that that is an alcohol. Same here. Phosphate group, it's just the phosphate ion. That is a functional group that is attached to a carbon chain or just an organic molecule. In this case, that's the pentose sugar, and it's what basically distinguishes it. And the phosphate group is just the anion PO43- in phosphoric acid, H3PO4. If you remember again from chemistry that acids contain hydrogen ions and then whatever anion it may be. And that is exactly why we call DNA or any other nucleic acid an acid, because it contains this remainder from phosphoric acid. If you can see, we have hydrogen ions attached to the oxygen, and they are the ones that give acids their very distinctive acidic qualities and properties. Anyway, you don't need to know that for A-level biology, not at all. That's just to help you understand if you're a little bit confused. Let's now take a look at DNA on a larger scale. And as you know already, it is just these nucleotides, one after the other, to form a very, very long chain. And they're linked together by these phosphodiester bonds. Here they are. So it's just a bond, it's kind of like a bridge between one carbon atom on one nucleotide and a different carbon atom on a different one. So we have an oxygen atom, phosphorus, and then another oxygen atom, and all of that makes this link. It joins together these nucleotides between the carbon atoms on the pentose sugar. Now you know that these nucleic acids are a very, very long chain, and the two ends, so the very ends of each the very ends of the chain, they are different from each other. They differ by what is found at the end of them, quite simply. And to distinguish between the two ends that are quite different, we say that one end is the 5' end and the other end is the 3' end. If you remember back from carbohydrates again, we learned how to number the carbon atoms on a monosaccharide, starting with the one closest to the oxygen, carbon number 1, and then we work anti-clockwise round, carbon number 1, number 2, number 3, number 4, and so on. And a shorthand way of saying carbon number 1 is just 1'. So therefore 5' is carbon number 5, 3' is carbon number 3. If you have noticed already, the phosphodiester bond is always between carbon 5 and carbon 3. At the very top of the chain, at the very, very top, if you can imagine, the very last nucleotide at the top, carbon number 5, so that's the carbon that's at the top, it is attached to a phosphate group. That's the shape of the end on that side of the strand. If we go all the way back down to the bottom on the opposite end, the very last carbon atom isn't attached to a phosphate group because there is no need to form another bond. There is no one else to form this phosphodiester bond with. There are no more nucleotides. It's the very last one, so therefore no bond needs to be formed. So it's basically just the pentose sugar on its own. And what is left there is, of course, just the OH group there, the hydroxyl group. So basically the 3' end is OH, 5' end is the phosphate group, and they of course differ from each other, and to distinguish between them we call one 5' and the other 3'. Okay, now let's talk about the DNA structure in a bit more detail. DNA actually consists of two polynucleotide strands, so not just one of these chains, but two of them. And they twist together to form a very specific shape that's called a double helix. We've seen that at GCSE already. Now what's important is that these two strands, they run in opposite direction. One runs from the 3' end to the 5' end, and the other one runs from the 5' end to the 3' end. And that is referred to as anti-parallel. We say that the two strands are anti-parallel. One goes in one way, one goes in the other way. Now the two strands, they are also joined together, and they're joined between the two bases. We saw those nitrogenous bases, yes, and they are joined together on opposite sides of the DNA molecule together through something called complementary base pairing. The bases on one strand are joined to the bases on the other strand through hydrogen bonds. And how exactly this happens, let's take a look right now. So let's take a closer look at these bases, and we divide them into two categories, pyrimidines and purines, purely by their structure. As you can probably guess from the diagrams, pyrimidines are the smaller ones, they have only one carbon ring. The purines are larger therefore and have two carbon rings. And the specific names for each of the four of the bases that are possible in DNA are thymine, adenine, cytosine, and guanine. And this is where the A, T, C, and G abbreviations are derived from. Once again, thymine, adenine, cytosine, and guanine. These are the four bases that can be found in a DNA molecule. One of these attaches to a pentose sugar, which in DNA is deoxyribose, and to a phosphate group. Therefore, there are four possible nucleotides. As we've said already, the bases are joined together by hydrogen bonds. There are 3 hydrogen bonds between cytosine and guanine, and 2 hydrogen bonds between thymine and adenine. This means that thymine always pairs with adenine and cytosine always pairs with guanine, and no other combination is possible. Thymine always with adenine, cytosine always with guanine, just because of the way these hydrogen bonds can form. And this is what we refer to as complementary. That's what I said previously. So let's look now at the DNA molecule a bit closer, and we can see that we have our double helix here. Each strand has these nucleotides, and the nucleotides are joined together between the bases using these hydrogen bonds via complementary base pairing as shown on the diagram. And what this actually is, it's just these two strands, each with their bases on the inner side of the DNA molecule. They're joined together by hydrogen bonds, and this means that overall the two strands are also joined together by these hydrogen bond bridges, inverted commas. So we have to know that DNA is only one nucleic acid. The other one is RNA. And the difference is the pentose sugar in DNA is deoxyribose, and DNA is the abbreviation of deoxyribonucleic acid. RNA is ribonucleic acid because the pentose sugar is ribose. And the only difference between the two sugars is that deoxyribose has one less oxygen atom, as shown here on the diagram. That is it. There are other structural differences between RNA and DNA. Let's have a look at a few of them. First of all, in RNA, thymine does not exist as a base. Instead, we have uracil. So uracil is also a pyrimidine, and it also bonds only with adenine, but just thymine doesn't exist, so it's replaced with uracil. So the four possible bases in RNA are A, U, C, and G. Okay? RNA is single-stranded and it does not twist, therefore. As we've said already, the pentose sugar is ribose rather than deoxyribose. And just as an extra little note, RNA has some variants, so to speak, and they are called mRNA, tRNA, rRNA, and some others as well. And we'll see why these are so significant in future videos. So once again, just to summarize the differences between DNA and RNA, DNA is double-stranded, RNA has only one strand, it's single-stranded. The pentose sugar in DNA is deoxyribose, that's why it's called DNA. RNA is therefore ribose. The four possible bases in DNA are A, T, C, and G. In RNA, they are A, U, C, and G. So thymine is replaced by uracil in RNA. And DNA does form a double helix shape. RNA is not twisted. And I also have to add that RNA is so much smaller than DNA. If I told you at the start of the video that there are around anywhere between 10,000 to millions of nucleotides on one DNA strand, well, in RNA there are anywhere between 75, only 75, and up to a maximum of around 10,000. So it's quite a major difference really in terms of length. That is it for today's lesson. If you have any questions, please ask in the comments. I hope this all makes sense. Next time we'll continue looking at nucleic acids, specifically at how they replicate. So I'll see you then. Thank you very much for your attention. Thank you for liking and subscribing. See you next time. Goodbye.